Hey guys, welcome back. This is our first set of notes here for Chapter 11, How Cells Reproduce. You could essentially call this mitosis. If you recall from general biology, IPMAT, IPMAT, interphase, prophase, metaphase, anaphase, telophase, that's what most of this chapter is really going to be about. It's going to be important that you understand what we're talking about today because this is really the foundation for the onion root lab that we're going to spend so much time performing this quarter. So let's jump right into it. Cellular reproduction. We talk about HeLa cells in the very beginning of this chapter. What we got is this cool story. A lady named Henrietta Lacks. She died of cancer in 1951, and she was only 31 years old. But they kept some of her cells, and they called them HeLa cells because it's the first two letters of her first and last name. And those cells continued to grow and continued to grow and continued to divide. And we still have some of those growing in laboratories today. And what we use them for is to investigate cancer, to investigate how viruses grow, and a lot of other processes in medicine and research because these cells never stop dividing, and because they're cancerous cells. And I think in your textbook, you check and they'll have a picture of a karyotype from all the chromosomes in some of those HeLa cells, and they're pretty crazy karyotypes. So please check that out. So what we can do from this is it helps us to understand the structures and the mechanisms that cells use to divide, and that helps us understand why cancer cells are immortal and why we are not immortal. And I think when you see the results of these cancer cells, you'll understand that constant division is by no means a good thing. And so here we have the picture of Henrietta Lacks and a picture of a few of her cells in the process of division. Some very cool photos. And that's why we use HeLa cells. So when we go to divide a eukaryotic cell, so a cell with a nucleus, membrane-bound organelles, that kind of stuff, we're typically going to do this in two steps. The first step is nuclear division, which we pay a lot of attention to, and then we have cytoplasmic division as well. So the sequence of stages through which a cell passes during its lifetime, the whole thing is called the cell cycle. I told you earlier, IPMAT, interphase, prophase, metaphase, anaphase, telophase, all of those are the cell cycle. And so it's basically the cell's birth to the cell's division is kind of the cell cycle. And so it is three phases: interphase, and then mitosis, which is prophase, metaphase, anaphase, telophase, and then finally cytoplasmic division, which really doesn't get that much attention, but this is actually pretty cool because we put all of our attention, of course, on the nucleus, but we have to divide rough ER, we have to divide smooth ER, we have to divide mitochondria, we have to move them all to each side, and so cytoplasmic division in and of itself is pretty complicated, but in your general biology courses, we spend a lot of time talking about mitosis because the genetic material is a thing. You really don't want to mess it up. So it gets a lot of time. Anyways, that's the process we go from one cell division to the next cell division. And so here's what it looks like. Cell's going to spend about 90% of its time in interphase. So we spend so much attention on mitosis, but really that's a teeny tiny piece of a cell's life. So the G1 phase, we have growing. The S phase stands for synthesis, and we are going to duplicate all of our DNA. The G2 phase is the cell preparing to divide, getting organelles where they need to be. And then mitosis is actually dividing our nuclear material, our DNA, and that's the prophase, metaphase, anaphase, telophase. And this little piece here called cytokinesis, which is cytoplasmic division. And then we are back into interphase, and the cell just being a cell. And so we used to actually call this whole interphase the resting stage because we didn't think the cell was really doing anything, and now we know that it's actually doing a lot. And I like this diagram because they have these cool little yellow circles for checkpoints. And so they have these built-in checkpoints right here that'll stop the cell cycle from proceeding until certain conditions are met. So we make sure that the cell is big enough, and it has enough organelles. We make sure the cell has replicated all of its DNA. We make sure that the cell has spindle fibers that are each attached to a chromosome. And if your cell fails any one of these checkpoints, we might stop mitosis, we might stop cellular division, we might wait until the cell has everything back in order, or we might trigger the cell to die. And so it's a pretty well-regulated process getting a cell to divide. So let's start with interphase. Interphase consists of 3 stages, like we just said, G1, S, G2. During this time, the cell is going to grow. It's going to double the number of its cytoplasmic components, so mitochondria, rough ER, smooth ER, lysosomes, peroxisomes. If it's a plant cell, certainly chloroplasts, and it's going to replicate its DNA during the S phase. And so G1, we grow. S, we double our DNA. G2, we get ready to divide. And most of your cell activities take place during G1. The G1 is really just the cell being a cell. And if it's a liver cell or a skin cell, it's doing whatever a liver cell and a skin cell will do. And the control mechanisms work at certain points in our cell cycle, which is our checkpoints, and we showed you those in that diagram. Some checkpoints will keep a cell in G1 and say, we don't need you to divide right now, so just keep doing your thing. And that's an important thing to have the ability to do. And if you lose control of these checkpoints, or perhaps if the cells are not obedient to these checkpoints, then we can have a cell that will die, which is okay because we can replace him, or we'll have a cell that is disobedient to the checkpoints and divides anyway, and then you can develop cancer as a result of that. So checkpoints are important. So mitosis can be useful for asexual reproduction as well. So mitosis is just this nuclear division piece, and it maintains the same exact identical chromosome number in both daughter cells. So our target here is to make clones. So in our multi-celled species, like you and me and dogs and flowers and cows and fish, our cytoplasmic division and our mitosis just increase the number of cells during our development. They will also replace our damaged cells later in life. So, you get a cut, it's not a big deal. New cells fill in. You grow from a teeny little kiddo to an adult, and cells are dividing and you're getting taller, right? Now, we can also use mitosis for asexual reproduction. So this doesn't quite work for you and I, but if you were a teeny tiny little unicellular, maybe like a yeast or something, sure, yeast, bacterium, something like that, a single individual can reproduce by mitosis and cytoplasmic division. So just kind of pinching itself in half and having 2 copies. So that'd be kind of cool if you were just one cell. But since we're made of many cells, we can't do that. So when you start to duplicate your chromosomes, you get to this cool word homologous. Homolog means the same. So pairs of chromosomes that are homologous have the same length, the same shape, and the same genes. Typically your homologous chromosomes, you have one from your mother and the other from your father. You will frequently hear that humans have 23 pairs of homologous chromosomes, and so you have chromosome 1 from your mom, and you have chromosome 1 from your dad. Those are homologous chromosomes that carry very, very similar information. And you have chromosome 2 from your mom, and you have chromosome 2 from your dad. And that goes all the way up to 22. And then you have sex chromosomes for 23, XY if you're a male, XX if you're a female, and that's how we get to our 46. So the only differences in a human genome is our sex chromosomes, but all of our autosomes are going to be homologous. And so we all start off with homologous chromosomes prior to replication. And so here we have our cell that is in G1. We've got, let's say, dad's chromosome number, 5. Why not? Mom's chromosome number 5. Okay? We get to G2. We've gone through the S phase. And in the S phase, we duplicated our DNA. So we're still in interphase. We have 2 copies of dad's chromosome 5 and 2 copies of mom's chromosome 5. We don't have all 46 chromosomes in here, or it would be a really complicated image. And then after mitosis, we make 2 daughter cells that are identical. This one has one of dad's homologous chromosomes and one of mom's. This has one of dad's, one of mom's. And so we made a clone. We have the same exact thing here as we do here and here. We made a copy. That's all we did. And what we're going to do is take a look at how we make those copies. So, how does mitosis maintain the same chromosome number faithfully? Well, the first thing we need to do is the pair of homologous chromosomes in G1. Both of them are unduplicated. They're just one copy each. By G2, we duplicate them. And then at the very end, after division, one copy of each chromosome is in each new cell. So from general biology, do you remember how we ensure that we get one copy of each chromosome in each new cell? Hopefully, you remembered we had these really cool structures called spindle fibers and centromeres. And the spindle fibers will grab the centromeres of each sister chromatid. That's a sister chromatid. That's a sister chromatid. And pull them to opposite sides of the cell. And so that's a big focus for us in mitosis, is how exactly do we maintain very faithful to maintaining the perfect number of chromosomes in each cell. And spindle fibers are the reason we're going to be able to do that. So a cell's not going to be able to do its job unless it has a full complement of DNA. We can't just drop a chromosome and be okay. It doesn't really work that way. Each cell needs to have one copy of each chromosome. Each chromosome contains many genes. Many genes contain the code for many different protein products, and we need those to survive. And so the cell replicates its DNA in preparation for mitosis, and we do our very best not to make any errors when we do this. And so our 4 stages of mitosis are going to parcel these sister chromatids into separate nuclei, and these phases are prophase, metaphase, anaphase, telophase, in that order. At the very end, we divide our cytoplasm, and we get 2 diploid cells. And that's important to understand because in the next chapter, we're going to look at how we get haploid cells. Diploid cells means we are maintaining these homologous chromosomes. You still have one copy from dad, one copy from mom when it's all said and done. The other way to call diploid is, let's see if I can write on my computer, 2N. That's the same thing as diploid, is 2N, which means you have 2 copies of each chromosome. And so each cell needs to have the same number and the same kind of chromosomes as the parent, because that's what it means to be a clone. All right, so we will stop there for now, and we'll jump back into how we control the cell cycle next time. So thanks for your time.